Hi everyone, my name's Leigh Ellis and I'm currently an Associate Professor in the Department of Medicine and Scientific Director in the Center of Urologic Research Excellence at the Samuel Ocean Comprehensive Cancer Center at Cedars-Sinai Medical Center in Los Angeles. I'd like to thank the organisers of today's forum for allowing me to speak to you today and share some of our latest work from the laboratory. So my lab focuses in on prostate cancer and more so how EZH2, a known epigenetic regulator involved in prostate cancer, drives both progression and therapeutic resistance. Now, EZH2 is the catalytic subunit of the polycomb repressive complex 2, where it catalyzes the trimethylation of the K27 or lysine 27 residue of the histone H3 tail. Now, functionally, this usually causes gene silencing by the formation of heterochromatin. But as studies have gone on over the years, we now know that EZH2, along like other epigenetic modifiers or these enzymes, can actually target non-histone targets. And some examples here for EZH2 are listed with STAT3. It can also act as a co-activator for transcription factors like the androgen receptor or NF-kappa B complexes, as well as work in methylation-independent fashion with regulating cyclin D and Wnt target genes. Now, over the years, EZH2 and the polycomb complex has been shown to be really important and a master regulator almost of multiple biological pathways and cellular processes, both in developmental biology and cancer biology, and some of those are listed here. And my lab, as I said, focuses both in on cell identity and immunity. Further, we've also seen the development of these catalytic inhibitors of EZH2 coming over the years, and they've come to clinical trials. And most recently, the Epizyme compound, EPZ6438, has just reached and received FDA approval, which is exciting news for the EZH2 inhibitor field. Also within our work, you'll see that I've used a compound tool more for experimental studies, DZNEP, which is considered a non-catalytic inhibitor. And so when it comes to prostate cancer and its overall response to immunotherapy in the context of checkpoint blockade therapy, we haven't really been doing so well. And what we're representing here on this graph is prostate cancer, where it is more represented by the trials conducted in the metastatic castration-resistant space. And so here you can see, just demonstrating briefly, that firstly, we know that prostate cancer doesn't come with a lot of somatic mutations or tumor mutational burden compared to other cancers such as melanoma or colorectal cancer. But we also have a very low overall response rate when it comes to the clinical trial outcome, just coming in just under 10%. And so efforts here from a combination standpoint are really proceeding the field as to how we can make prostate cancer more responsive to immunotherapies or checkpoint blockade therapies. Now, the area where my lab is most concerned is about therapies in the context of prevention of high-grade, more aggressive primary prostate cancer and preventing them from progressing to castration-resistant metastatic disease, which is often lethal for the patient. And so this nice summary within this Science article a couple of years ago clearly demonstrates with these two major parameters of measuring the hotness of cancers, both by tumor mutational burden and gene expression profiling, which is more around an interferon gamma or interferon signature, otherwise known as a T-cell inflamed signature. You can see that prostate cancer is pretty negative for both of these readouts, indicating that especially in the primary setting, that prostate cancer doesn't have a lot of immunoreactivity going on and is potentially, as I said, one of the coldest landscapes that we're dealing with in the cancer disease spectrum. Now, another term that I want to introduce to you here and where our work has led us is the context of viral mimicry and how this has been induced by epigenetic therapy in cancers. And I've just put up a couple of examples here of sort of leading papers that have come out within the last couple of years. More so the paper from Daniel Di Cavello up in Toronto that sort of led this field, first publishing in 2015, this Cell paper. And really what these papers demonstrate to us is that by the use of epigenetic therapies, that we've been able to modulate or reprogram the tumor and also its tumor microenvironment to make it more conducive to response to checkpoint blockade therapy. So when it comes to epigenetic therapy and the induction of viral mimicry, really what I'm explaining, and this is more the story from our work, there are other versions of this, so this is just not a one-size-fits-all. But basically, through the use of epigenetic therapies, in our context, EZH2 inhibition, it results in chromatin modification and the re-expression of double-strand RNA or repeat elements. We focus in on a subset of these double-strand RNA species known as endogenous retroviruses. And what these double-strand RNA species will do is relocate to the cytoplasm, where they're then detected by these sensors known as RIG-I and STING. These are the two examples I'm showing you here. And with that, these sensors then signal usually to STAT1s or STAT heterodimerization, along with dimerization with ERF proteins to then transcribe interferon stimulated genes and provide the host with a defense against what it thinks is a virus attack. Now, within these interferon stimulated genes, and just again representing some of the genes that we see and also other studies, is we get some important genes coming into proteins here, where we get the chemokines CXCL9 and CXCL11. These are chemoattractants for T cells. We also get antigen presentation machinery in the context of beta-2M and HLA-A, just two genes shown here. These are important for MHC class 1 cell presentation of antigen to the microenvironment. And also we get the brakes of the immune system here, where tumor cells will often express PD-L1 to evade the immune system, but also in other forms of immunology, these are also brakes of the system where they will bind to the T cell receptor PD-1 and ultimately shut that T cell down and make it exhausted. So really, again, just what we're seeing here through this mechanism is upregulation of immune recognition, of the tumor cell and immune recognition, the context of increased immune trafficking or the potential to increase immune trafficking, but also the increasing the brakes of the system or immune suppression. So this all started really in the lab when we were looking at, in the context of EZH2 being a synthetic lethal interaction with the overexpression of c-Myc, which is a known driver of prostate cancer. And we thought that if we could target EZH2 early, it may be a vulnerability in primary prostate cancer to ultimately shut it down and make it a disease where the patient does not have to worry about progressing to metastatic state and then dealing with the forms of androgen deprivation that they'll be treated with, and ultimately coming that therapy-resistant lethal prostate cancer. So what we found, and firstly just introducing the mouse model that we used, where we have the high c-Myc overexpressed in the prostate cancer luminal epithelium, and then we introduce floxed alleles of EZH2, and basically by using a probasin Cre recombinase also in the mouse, we were able to exclusively cut out the set domain of EZH2, making it catalytic dead, ultimately affecting or shutting down its catalytic function. And so what you can see in organoids from the mouse here, and we also have this represented in the gems themselves, but we just see the growth over 8 days here of these organoids. And then what we saw is we saw it more just a delay of the growth rather than an actual stoppage, which was ultimately, surprising and both sort of not what we wanted to see, based on our original hypothesis, seeing that this redundancy had come in and eventually these organoids would regrow and join their wild type counterparts. So we came in at day 3 where these growth lines were quite separated, and we applied RNA sequencing to see if we could just sort of see anything that could indicate an escape route for these cancers or these organoids at the time. And so what we found firstly is when we did both chemical inhibition of EZH2 with DZNEP or the genetic modification, where this is a tamoxifen inducible system, so that's why you see tamoxifen here to elicit the EZH2 catalytic domain deletion. We can see that we had differentially expressed genes. So genes were going up and down as we expected, but we also had some important observations here where we were getting loss of the canonical function of EZH2 here, both with loss of the K27 trimethyl mark, and also we were inhibiting the cells by lowering the EDU incorporation into these cells. And this was more of a cell proliferative inhibition rather than a cell death mechanism. So we performed GSEA on our RNA sets, and to our surprise, we enriched for innate immune genes, or basically interferon alpha and interferon gamma signaling pathways, both in the genetic and the chemical data sets. We then used our DZNEP, work that I'm not showing here, but it is in the paper. Using our DZNEP gene expression data, we derived an EZH2 activity score to where we could then rank patients based on EZH2 activity. So doing that, we then quartile separated these patients, and we compared quartile 1 with quartile 4, the bookends, the highest and the lowest activity scores, to then do the same analysis through gene expression and GSEA. And just showing you 2 data sets here, primary data where we have laser captured microdissected prostatectomies here from an NCI cohort, and also the TCGA publicly available data set. And when we did this, again, we saw that the interferon type 1 and 2 alpha and gamma signatures were enriched in the NCI data set, and the same for the TCGA, with gamma being the most enriched and alpha being somewhat down, but still very significant. So we overlapped both our data sets here from our initial mouse experiments, just to see what was shared and what wasn't from a potential master regulator perspective. And while we had a good number of interferon related genes, we also had other drivers of other processes, including EMT and some DNA damage. But first and foremost was the interferon signaling. So taking into account all of our data sets that we used, and I showed you 4 of those, the one that we did add here was a castration-resistant cohort from Misha Beltran's data. These are just the adenocarcinomas, which is what's represented here, a full adenocarcinoma data set. And by looking at these and looking for an overlap of at least 3 out of 5 data sets, we derived a 97 gene set that, as you can see, is nicely enriched for interferon immune-related genes. Now, using these 97 genes, we then went to a set of prostatectomies in collaboration with Matt Friedman at the Dana-Farber Cancer Institute, who had done a lot of epigenetic profiling within these patient samples, and these samples can be found in a recent Nature Genetics paper from his lab. And we were expecting these genes just to be really repressed by the enrichment of a H3K27 trimethyl peak when we looked at the ChIP-seq data. And to our surprise again, we actually found that these genes were actually not associated with K27 trimethylation at all, indicating that something else was being directly affected by the EZH2 inhibition to then enrich for this gene signature. So other data sets that we were able to use from these patients was both K27, H3K27 acetylation and also ATAC sequencing. So K27 acetylation is a mark of active chromatin or active transcription, and ATAC sequencing is when you see a peak, you have open chromatin or euchromatin, whereas if a peak is not there, you have heterochromatin or the chromatin is compressed. And funny enough, what we did see here was that the majority of our genes were associated with an active mark, K27 acetylation, and also open chromatin with the positive ATAC peaks here that we saw. So we were deeming these genes to actually be primed for activation rather than repressed and going through the full chromatin remodeling mechanism to then activate them. And when you think about these genes as predominantly being a defense mechanism, that sort of made sense to us. So coming back to the viral mimicry, we sort of thought that we must be inducing viral mimicry to then activate indirectly these interferon stimulated genes. And so going into two organoid models here, these are our mouse organoids from the MIC model that I showed you previously, and then a human prostatectomy that we were able to get within as a short-term culture in 3D, also in the lab, and then treating them with the two EZH2 inhibitors and interferon gamma as a control. We showed nicely here that we were able to upregulate drastically the double strand, the total cellular double-strand RNA levels. Looking in vivo to where we transplanted our high MIC 2D cell lines into mice, black 6 animals here, and then treated with the EZH2 inhibitor. We also showed that this treatment resulted in vivo of an upregulation of double-strand RNA, giving us the context that we were indeed inducing double-strand RNA species at the cellular level and potentially invoking a viral mimicry response. We wanted to sort of prove this a little bit more, and so we went to a data set that was published by Steve Bulk and Wei Yi, that at the time Wei was at Beth Israel in Boston. She's now at UCLA. And she had published a story on prostatectomies looking at PD-L1 foci. And so she had a subset of prostatectomies here that were either PD-L1 high or PD-L1 low. And now PD-L1 was one of our interferon stimulated genes of interest. And I'll get to some of that data in a minute. But basically using this data set, and the premise of what I was just talking about, we expected to have an inverse correlation here with both K27 trimethylation and also double-strand RNA cellular levels. And again, these are just untreated tissues, so this is just baseline levels that we're looking at here. And so what we saw is that in the PD-L1 low tumors, and where we expected high K27 with low double-strand RNA, this is what we exactly saw. And then with a PD-L1 high tumor set, we saw the inverse, where K27 trimethylation was low and double-strand RNA was high. So this may suggest that there are potentially prostate cancer patients out there that could have a hot tumor and potentially responsive to checkpoint blockade therapy, or the fact that we just don't have enough heat, if you will, switched on. And so by using epigenetic therapy, we may positively affect both of these types of populations. So trying to find out where our whole viral mimicry or double-strand RNA species were coming from, we again, and unfortunately this actually was a question from a reviewer at the time, and under COVID conditions, we couldn't actually enter the lab and do the experiments, but lucky enough, there was a data set out there from Jindan Yu's lab in Chicago, where she had used an LNCap human prostate cancer cell line, and she used hairpins to knock down EZH2 versus the hairpin control. And so what we firstly saw here, which was a nice control compared to the prostatectomy samples that I showed you earlier, is that our 97 gene list of interferon stimulated genes was not associated with any enrichment of the K27 trimethyl mark, but again, they were mostly associated with a K27 acetylation or active mark. Upon EZH2 knockdown, we got more enrichment or an increased enrichment, which went along with what we're describing as activation or transcriptional activation following EZH2 inhibition. Now, in collaboration with David LeBay, a friend from, we both met at Dana-Farber, he's now running his own lab up in Montreal. He used a bioinformatics pipeline that he has in his lab, where basically we wanted to use this ChIP-seq data to ask genes that had endogenous retroviruses housed in their 3' UTR, and what genes of those genes lost K27 trimethylation with a concurrent gain of K27 acetylation at the time of EZH2 knockdown. We came to a conclusion of 302 genes that were under this cutoff, and those are actually in the supplement data of our paper. And just showing you the summation of this as to where we take these 302 genes and we look here to where we get a loss of K27 trimethyl here in the blue line following EZH2 knockdown, with an overall concurrent gain of K27 acetyl indicating activation. So this all fit the bill that we were indirectly targeting these genes, or sorry, I should say directly targeting these genes to induce a viral mimicry response through the release of 3' UTR housed endogenous retroviruses, and thus indirectly getting the interferon stimulated genes that were enriched in our original RNA-seq data. So coming to this, we sort of wanted to work out mechanistically who was a middleman. We focused in on STING just because of a potential therapeutic interest with STING. And then when we looked at our data sets, just correlation data here of STING expression against our EZH2 activity score, we can actually see here that the activity score is from left to right, EZH2 high activity to EZH2 low activity. And so as you can see, as the activity becomes lower from left to right, we get more STING expression. So we had a positive correlation here with low activity and overall STING expression. We also saw the same for RIG-I and some other sensors in this family. Now, functionally, we came in with two mouse cell lines that we use in the lab, prostate cancer cell lines that are derived from the genetically engineered mouse models where P10 is knocked out, or as I've already introduced, c-Myc is overexpressed. Now, these are on a black 6 background, which will be important for another experiment coming up. But as you can see here, when we performed chemical inhibition of STING, we didn't lose the induction of double-strand RNA. So STING is obviously downstream of the double-strand RNA because it has to sense it, so that made sense. But what we lost was two critical interferon-stimulated genes. Now, these are by flow cytometry, so this is protein levels here. But we lost tumor expression of PDL1 and also the potential for tumor expression of MHC class 1. So again, preventing the potential for antigen presentation. We also doubled up on this with genetic validation of STING using CRISPR. And when we CRISPR'd out STING of these cells, we basically saw the same response as you can see here. So STING was an important mediator for this, for the cell to upregulate interferon-stimulated genes. So in vivo, and again using our c-Myc, high-Myc transplant model in black 6 mice, you can firstly see that our tumors here are both resistant to PD-1 antibody, and the epizyme compound, the EZH2 inhibitor. Now, these tumors grow quite rapidly, as you can see, as we reach an ethical endpoint here of around 18 to 20 days, usually. But when we combined the two therapies, we could see that we got quite a significant slowdown of tumor growth. Now, this wasn't a stop, but slowing this tumor down the way that we did was a pretty big statement for this model. So we were very happy with this. Having a focus on tumor PD-L1 levels at the time of this study, we also performed on our tumors, we did CD45 sorting, and on our CD45 negative cells, which is basically the tumor cells, we could see that the tumor cells did have an upregulation of PD-L1 in response to the epigenetic therapy or EZH2 inhibition. We also confirmed this in our two organoid models, the top level being transcription here. So with both EZH2 inhibitors, we got a nice transcriptional upregulation as expected, as it was one of the interferon stimulated genes in our data set, and at the protein level as well, where we got quite a nice upregulation in both our systems. So with PD-L1 upregulation, we wanted to functionally validate this as well. And this is where the strain of the cells, the mouse cells that we use here, are critical. Again, our prostate cancer models are from a Black 6 strain, and this is a cytotoxic assay where we rely on an MHC class mismatch and an immune cytotoxicity by incubating them or co-incubating them with FVB splenocytes. So just to give you an indication here, our background cytotoxicity after 4 hours of co-incubation is represented here by the white bars. Now, when we pre-treat these tumor cells with EZH2 inhibitor and upregulating PD-L1, we can see that we induce a protective protection against the cytotoxicity here. And then when we introduce an antibody, a PD-1 antibody, we break this protection and we get back or rescue our cytotoxicity of the splenocytes, or induced by the splenocytes. When we CRISPR out PD-L1 in these cells, we can see that we ablate all this. There's no protection here represented by these two bars, and again, no significant increase or recapturing the cytotoxicity as expected. We again validated this in our second model, the P10 knockout model, again on a Black 6 background and incubated with FVB splenocytes. Again, our control kill here, once co-incubated, we see the protection and we see the rescue of cytotoxicity. And again, CRISPRing out PD-L1 in these cells, we also ablate that, showing this is PD-L1 dependent protection here that we are inducing with EZH2 inhibition. So just to give you an idea of just the tumor microenvironment, and I'm really just showing here the two biggest sort of movers were both our T cell populations, where we increased our overall tumor infiltrate, both with EZH2 and combo, more so with CD4 and CD8 cells. We also moved and reprogrammed, we think, the tumor-associated macrophages, where we had iNOS positive M1 TAMs here being increased by both combo and EPZ, EZH2 inhibition. Overall, we get down to combo, we get our biggest loss of ARG1 positive M2 TAMs, and of course the ratio here is greater M1 to M2 in our combo. So we think we're getting a little change in the paradigm there, with suppressing our immunosuppressive TAMs and gaining our cytotoxic T cells with our anti-tumor TAMs as well, the M1 TAMs. Now, looking at the CD4, CD8 subsets, we can see here that we don't actually activate here, looking at 41BB as an activation marker of T cells. We can see that we don't affect the CD4 positive cells, but we also don't see a PD1 shift on these cells. But in the CD8 population, as expected with a PD1 inhibitor, we do see activation of these cells both in PD1 and combo arms, and also EZH2 inhibition does downregulate, not significant, but downregulates PD1 on these T cells. So that overall just gives you a summary of the story. Now, what's exciting, we've just been funded by Epizyme to conduct a phase 2 trial, a neoadjuvant study where we will pre-treat patients with the EZH2 inhibitor before prostatectomy. And this is to provide a proof of concept that indeed EZH2 inhibition can heat up the prostate tumors adequately to where we can then hopefully move on to a combination study in patients. So I'll just point out these two papers for the full story, published last December and in March of this year. This is just a summary of this work, but these two papers give you the full story. And just want to thank really an army of people here that have helped me over the years, but also helped me with my recent move to Cedars-Sinai from Dana-Farber. And also my new lab members, and also Catherine and Debbie, who really spearheaded a lot of the work. Catherine's a postdoc, Debbie's a scientist, but these two ladies really spearheaded a lot of the work that's coming out of my lab as of late. And so a big thanks to all them, the fellow faculty and also the collaborators. And thank you again for your attention, and happy to take any questions.